Okay, welcome to a lecture about food science and some of the microorganisms that are involved. And I don't know, I just like talking about food mostly. So we're going to talk about food, we're going to talk about history of food, and we're going to talk about what food science is and then some specific organisms. So to get started, this is a quote from the Institute of Food Technologists. And it defines food science as the discipline in which the engineering, biological, and physical sciences are used to study the nature of foods, the causes of deterioration, and the underlying principles of food processing, and the improvement of foods for the consuming public. So that's pretty clear. Essentially, food science is part biochemistry, part nutrition, part horticulture and agriculture, part engineering and microbiology, all sort of rolled into one. There's food economics that is even part of this. Food science is a big, wide category, and whatever your personal interests are related to food, you'll find that there's a part of food science that you can fit into. That said, it's typically understanding sort of the pre- and post-harvest issues, as well as food processing issues in industrial food and things like that, as well as the microbiological processes that create the food that we eat, as well as the processes that break down the food that we eat before it gets to our plate. I don't talk a lot in this about post-harvest, and post-harvest physiology is a big part of horticulture, but post-harvest microbiology and post-harvest chemistry is a big part of food science as well. And the engineering, that can be whether you're engineering the foods or engineering systems to produce, to preserve, etc. the foods. So we're just going to talk about some of these foods and their history. I'm kind of in the way, but that's wine. Wine is at least since 6000 BC from Georgia across Persia and Italy. And wine is made from grapes, and it likely, we'll talk about it a little bit more, but all of these likely began to form from wild yeasts. Beer, even a little older than wine. It's found in China and Persia, and bread as well, central to kind of the organization of early human societies. I don't need to get into that because this isn't the scope of the class, but I'm going to talk about some foods though that I like, but also that I feel like are important or have interesting stories too. So fish sauce actually is one of those from the west to the east through the Silk Road type of foods where garum, this whole fish, some was made with whole fish, some wasn't, fermented fish product that the sauce was developed from. It arrived in China and Asia and was quickly adopted due to their local fishing, and was really the most popular condiment there until about the 14th century in China, where a different microorganism was used to ferment soy sauce, and then soy sauce became the predominant, particularly in China and Japan. Still today, fish sauce like that Squid Brand is a Thai sauce. We could give a whole lecture on, and it really wouldn't fit in this class, but on the different types of fish sauces. But essentially, they can be made from fish. Basically, fish sauce, they take a bunch of fish and a bunch of salt, and they put it in a barrel or a bucket or a container, and a fermentation process happens, and the liquid is pressed out, and from that there are different levels, the first pressing of the sauce and things like that. And soy sauce is made in a totally different process. Okay, so we can talk about fermentation. Fermentation, the conversion of sugar into ethyl alcohol by yeast and other microorganisms. Early beer and wine likely due to spontaneous fermentation by wild yeasts. You know, someone had some gruel or some porridge that was ground up wheat or barley in the corner, and they were eating from it, this paste, and it sat in a bowl, and they noticed in a day or two there were some bubbles, and they thought, I wonder what'll happen if I cook. This is just conjecture, who knows, you know, I wasn't there, but I wonder what'll happen. And they tried that. You know, there's fun stories about mead happening by a honeycomb falling in some stagnant water and someone drinking it and noticing it tasted different. You know, that sounds kind of fantasy. It was possibly due to something more like early beer and wine. You know, early beer could have been more like a thick gruel, and in fact I didn't put it on here, but some of the early beer is a thicker ceremonial gruel essentially. But anyways, this is that process. In seeing now, I can tell that I haven't, this picture is being blown up too big, but essentially I'm not going to get into the chemical process here, but it is conversion of sugar into alcohol, absent the presence of oxygen. It's part of the rare respiration. Something interesting, through fermentation, heme protein that kind of makes the Impossible Burger bleed is produced for that process through fermentation. And this is a short list. This is just off the top of my head, all the fermented foods I could think of. Kimchi, sauerkraut, pickles, beer, wine, yogurt, bread, buttermilk, fish sauce, soy sauce, gochujang, kombucha, miso, salami, Tabasco sauce, tempeh, vinegar. There's a comma, so I obviously wanted to keep going. I'm sure you could keep going. All of my favorite foods are right there, so, you know, let's just say something. Okay, so yeast. I've got some life cycles here. So Saccharomyces cerevisiae is the yeast behind bread, wine, and beer. And to go through this life cycle a little bit, so we've got let's start here. 4 haploid ascospores within an ascus. That ascus ruptures and the spores germinate. And now if you remember from Ascomycota, we've got those different mating types in that ascus. And so we've got vegetative growth of haploid cells, and they form hyphae, and then they actually can mate between those cells, and we can have a diploid stage as well. And that can have vegetative growth, and an ascus can form in the right situation, meiosis, and again, we can have the ascospores come. So there's sort of 2 or 3 separate circles depending on how you draw yeast. And this is a very, very basic version. I'll be candid, this isn't going to be one of the life cycles I wanted to test you on, but we gave you a much simpler Ascomycota to look at. Here's Aspergillus, just like a wild genus that has so many incredible species that influence our lives. Sake shows you rice vinegar, and then we've got miso and soy sauce are all fermented by members of Aspergillus. And notice here, we've got, in this life cycle, we've got, an ascus that forms, and ascospores. There can be a haploid, mycelial type of structure, and homokaryon actually, but we're not going to worry about that. And again, I'm not going to test you on this because again, it is complicated, but I wanted to show you here's some other types of life cycles that exist within Ascomycota. Okay, something else that happens in Aspergillus is, from a food science perspective, is these are found in legumes, tree nuts, and cereal grains. They're thermotolerant, meaning they're not killed when other fungi are killed, and they can be carcinogenic, they can damage the liver, they can make you very sick. And they're found, like I said, in peanut cultivation, Here we have them in corn. Those are pecans. They're a major economic problem because, particularly in Texas with the pecan industry and some other places, they can cause yield loss, as well as be something that if it's not caught, they can make you sick, and therefore there are limits placed when they're tested by governmental agencies. And so when you test the pecan crop, there can only be a certain amount of these aflatoxins that are produced, and it's the aflatoxins that are carcinogenic. Now there is AF36, which is a non-carcinogenic, non-aflatoxin producing strain of Aspergillus flavus, and that particular species, or that particular strain, outcompetes the strains that do produce aflatoxin and are carcinogenic. And those are used to inoculate pecan seed and other things so that when they go to grow, these outcompete them. And this is just like a human health, and it's sort of out of place in this food science, but I am talking about this genus. And again, this is Fumigatus, and it infects people who have a decreased immune system through their lungs or through their skin. As we see here, we can have an alveolar infection or through the vessels in the skin. And this kills 600,000 people a year with a 25 to 90% mortality rate. That mortality rate is both so variable and so high at one end due to the fact that it is sort of getting in on people with their compromised immune systems. And so a lot of times people will encounter this post-AIDS therapy, post-cancer treatment, post-surgery, and things like that. Finally, I just want to talk to you a little bit about careers in food science. Despite the fact that I was always jealous that my greenhouses were next to food science in grad school, and I got to smell all the delicious things they were working on, or there were classes where they went and they made wine and beer as part of their class. Besides that, there are all these great careers. Food inspector for checking levels and things like that. There's the industrial food where you're talking about large-scale factories producing food in industrial quantities. Product development, food microbiology, food engineering. This is just a tiny snapshot. Honestly, I highly recommend you look into food science as a potential career. Like I said, any of your personal interests within botany likely, or biology, likely have an overlap with the food science community. But that is it for today. Again, I just wanted to cursorily touch on some of the broad strokes of food science, as well as talk about a few of the microorganisms that fit into kind of what we're talking about in this class. Okay, I have some notes I made for review, and we've got a midterm coming up, and so there are some things I'm going to talk about here. I'm sure I'll forget some things. If you've got questions, send me an email. Beyond that, there will also be a Microsoft Teams meeting that Dr. Rotter hosts for both sections 1 and 2, where he will talk to all of you about really any questions that you have. But I've got some notes here that I'm going to go over just to make sure you know what's in my head as we're writing this test. So review all of the life cycles. You're going to need to be familiar with these life cycles. Okay, you're going to want to pay attention to the following things. You're going to want to watch ploidy. Is it haploid? Is it diploid? Is it where's plasmogamy and karyogamy taking place? Is it N plus N, 2N, N, those kind of things. And you're going to want to watch for, you're going to want to make sure you know those main structures, and ascus and basidium and things like that. You're going to just, any of the things that I labeled up here, any of the things that are labeled in your books, you're going to want to be familiar with those. That's going to be something that will help you a lot. Again, know the differences between Basidiomycota and Zygomycota and Ascomycota. from the videos that we talked about. Okay, when we talk about lichens, you're going to want to know crustose, foliose, etc. You're going to want to know those different forms. You need to know if they're photo or myco. You're going to need to know about mycorrhiza. All right, and endo versus ecto, the differences, some of those structures, Harding-net, things like that. You're going to want to know how they operate, how they're different, which ones are more prevalent. You need to be able to tell us what a plant is, to briefly describe photosynthesis, and you're going to need to talk about why we as botanists study things that aren't necessarily plants, and history of plants. We talked about that. Know those big broad strokes, those when major things happened. For my mycology, my growing mushrooms, the big thing I want you to take away from that is, why would I be telling you about growing mushrooms? Maybe you found it was interesting, maybe you didn't, but what I wanted you to get out of that is why the things work to grow mushrooms, and what kind of conditions we're replicating in the cultivation process, and just to get your head to thinking, okay, how are we copying the environmental conditions that exist in nature in order to cultivate mushrooms. There may be some questions about the specific processes, but mostly think of it in terms of the framework of this class, being a botany course, not being a horticulture class. So trying to think, that's, know what makes diatoms unique. That's all I can think of off the top of my head. But again, there will be a question and answer section, and that will happen after we've got the test fully written. I've got some questions put together and some ideas, so that's just some stuff there. But if you've got a question about what's on the test, or a question about something you're studying, you can send me an email and I'll answer it. So that's it.